And I would like to thank Laurent for inviting me to this meeting. It's always a pleasure to come here. I would also like to thank Shubham in my lab, who really did the groundwork for this project. More specifically, I would like to thank Shubham's foot, because Shubham's foot was instrumental to this work. I'll explain in a second. So Shubham started his internship in my lab last summer. And about 2 weeks into his internship, Shubham broke his foot. And which meant that for 6 weeks, he actually had to stay at home and he couldn't go to the lab. And this was at a time where I was looking for any sort of published material that would really give an overview of antiviral drug approvals over the last 2 or 3 decades. And I couldn't find anything really in the literature that was really what I was trying to find. So Shubham and I decided that actually that was going to be his new project while he was laying in bed at home on his computer. So thanks, Shubham, and thanks, Shubham's foot. Anybody recognizes this molecule? Sorry, I don't know if there's a lot of chemists in the room, but any guess? No? Okay. Yes, that's AZT. This is the first drug that was approved for HIV. In 1987, which is remarkable considering the virus itself was only identified in 4 years prior to that. And actually this is, when we started this project last year in 2017, we also had this idea that we were going to look at the 30 years of antiviral drug approval since the approval of AZT. So what we did with Shubham, and actually this is really Shubham's work, is to search databases for all drugs that have been approved in the US, so FDA approvals, and specifically focus on the ones that are antivirals that you can see. So Shubham searched different databases. I'm not going to go through the details on how those searches were done. Actually, Shubham has a poster, so if you have any question, with respect to how the search was done, or even how he broke his foot, Shubham will be by his poster, so feel free to ask. But the thing that was important for us is that we wanted to, let me see if I can find a laser pointer. We wanted to exclude things that were not direct antivirals, so things like PK enhancers or boosters. Also, we excluded vaccines because they were really, it's a different beast. We didn't really want to do the same analysis for vaccines. And really what we were mainly interested in are new molecular entities or new biological entities, as well as new combinations. So Shubham came with, at the end, about, I don't know, exactly 88 drugs that have been approved between 1987 and 2017. So what he did is he collected data with respect to generic and trade name, as well as the year and the month of approval, the indication, and things more related to mechanism of action. So for example, viral versus host targeting, as well as small versus large molecules. And large molecule is a big term. There's different kinds of large molecules. Also whether the drug was approved as a monotherapy or combination therapy, and other parameters. And this is really, in a nutshell, the result of Shubham's analysis. You see this, I would say, fairly linear progression in drug approval since '87 at a rate of about 2.8 approvals per year. Actually, when I say linear, it's not really true. If you look with a little more granularity, what you see is that we identified 2 peaks of approvals, one that is in the mid-90s, that is highlighted here, mid to late 90s, and another one that was really between 2014 and 2016, 2017. So we color-coded based on indication. So you see already it's pretty clear that there's a lot of drugs that have been approved for HIV. I'll get to the details in terms of the different viruses. This is also another way to look at the data. So you see the total approvals here per year, and you see the 2 peaks that I was referring to. And actually the first one coincides with a surge in HIV approvals that you see in the mid-90s, starting in 95. And the second one coincides with both HIV as well as HCV drugs here. And if you look at this as a pie chart, you see that half of the approvals roughly are an indication for HIV-1. And after that, you have hepatitis C virus, followed by HPV, herpes viruses, and influenza only 4 drugs. So another analysis that we did was to look at mono versus combination therapies, and what's really clear and also makes sense is that at the beginning you only have monotherapies, but then you see also in those 2 time points of peaks of drug approvals, you start to see more combination therapies. And so what we see is the result of maturation in clinical pipelines. So as you have more HIV drugs that are approved, they start to be tested and approved as combinations. And the same thing happened with HCV, except this time it was accelerated. A lot of the HCV drugs were only approved as combination and not as monotherapies. And we also were interested to look at the type of drugs that were approved, and you can see a majority of them were small molecules, so the typical sort of, I would say, traditional pharma research type of compounds. Early on, you had a number of large molecules. Those are mainly interferons. There was also one antibody that has been approved for RSV, one peptide fusion approved for HIV-1, and one antisense molecule that targets cytomegalovirus. So over the last 30 years, really, you see a majority of small molecules. Looking at also mode of action, you see a majority of compounds that target the virus itself and not the host. So host targeting compounds are a minority. And looking at the type of mechanism of action, the type of targets, what we found is that a majority of them were non-structural proteins within the viruses, so whether enzymes like polymerases, integrase, or protease, as well as HCV and S5A non-structural protein. So those actually represent about 3/4 of the drugs that have been approved over the last 30 years. So what this is telling us is that at least in the field of antiviral research, when it comes to clinical trials and approvals of drugs, what has worked in the majority of time are small molecules that target directly viral proteins. And as I mentioned before, mainly non-structural viral protein, which means that it's really intracellular part of the virus replications. The things that have been much more challenging is to take small molecules and target either the host or the immune system in a sort of like host-mediated antiviral effect. I'm not saying that it doesn't work, it's just been more difficult. Same thing, biologics or large molecules in general have struggled to get approved as antiviral drugs. But there's one nuance that I really want to point out, is that the thing that has worked really well, besides small molecules, is vaccines, obviously, which are different types of biologics that also target the virus through immune response. So, I mean, one of the things that I'm particularly interested in and that we were trying to convey when we did this project is the concept of clinical success rate, which is important in the pharmaceutical industry because in a way you're looking at the likelihood of approval of your drug when you start clinical trials. So the question is, what are the chances or how many of the drugs that enter clinical trials get eventually approved? And maybe it's something you've heard before, but on average across the pharmaceutical industry, you have about a 10% approval rate. So that means that 90% of the drugs that enter clinical trial fail for various reasons. What's really interesting is that, so if we compare, for example, by different disease areas, oncology is notorious to have a very low likelihood of approval. Again, there are multiple reasons for that. I don't think it's really the subject of this presentation. But what we were interested in trying to understand is how does infectious disease compare to the rest of the pharmaceutical industry? And actually, the likelihood of approval is about twice compared to average. And so there are multiple explanations for that. One that I favor, and I don't think I'm the only one, is actually has to do with the fact that when you use molecules that directly target the pathogen, in this case the virus, you actually, all your preclinical development is much more predictive of what you're expecting to see in the clinic. And if you look across the entire pharmaceutical industry, what you see is that it's exactly at this stage of first proof of concept efficacy that most drugs tend to fail. So in phase 2 clinical trial, when you start to look at efficacy of your drug, that's when you start to see effect or lack of effect If you look at infectious diseases and antivirals, that level is actually substantially higher. So that's the first part of the analysis that Shubham did. The second part of it was what we were really interesting to find out is if we compare drugs that have been approved versus antivirals that are currently in candidates in clinical trials, are we seeing changes in trends of everything that I showed you before? And so I make the comparison with the stock market with, just because things have worked in the past doesn't mean that those are the things that will work in the future. And this is something we wanted to find out. So what we did is that we looked at all major viruses and we broke it down chronic versus acute viral infection. And what you can see both in this graph and in this table is that there are still quite a large number of drugs that are in clinical trials for HIV, which is amazing considering how many drugs are already approved for HIV. Hepatitis B, the field is really exploding in clinical trials. So there's also a really large number of HPV drugs that are being tested. HCV, it's decreasing because actually now there's some really good treatment options. What's also remarkable is that there are a number of anti-RSV and anti-influenza, two respiratory viruses that are being tested with clinical candidates. So can we compare things that have been approved in the past with things that are being tested now, and are we seeing changes in trends? So if you look at virus versus host-mediated effect, you see actually a larger proportion of host mechanisms in clinical trials when you compare to approved antivirals. Also, we see a larger proportion of large molecules. So those could be, for example, monoclonal antibodies or other types of large molecules, RNA or oligonucleotides and others. Larger proportion compared to what have been approved in the past. And also for a number of reasons, you see a lot of monotherapies compared to a larger number of combination therapies in previous approvals. The main reason for that is that there's actually a number of first-in-class molecules or molecules that cannot be combined with anything because they're targeting a virus where there's really not a lot of treatment options. So I just wanted to highlight three of the main areas currently of clinical development of molecules that are either in phase 1 or phase 2 or 3 clinical trials. So as I mentioned before, I think HIV, there's a very significant amount of clinical research. The long-acting formulation is the idea of injecting drugs ideally every few months, maybe just twice a year during a doctor's visit. And so there's a number of patients that would like to switch from a daily oral treatment to this type of treatment options. So there's a number of clinical trials that are trying to figure out if that can be done. And then we heard those different approaches of latency reversal agents, the shock and kill or the kick and kill approach, sometimes even combined with broadly neutralizing antibodies. So we're seeing this right now being tested in the clinic. As I was telling you before, that HPV is a field that is exploding. So what we're finding is a number of new types of molecules that target the capsid, so they call the capsid assembly modulators. There also are a number of immune response targeting mechanisms such as TLR7 agonists, as well as S-antigen blockers. And finally, something that is also very significant in the field of HPV is a renaissance of antisense and siRNA, or things that target HPV messenger RNA in general. And finally, the other trend that we're observing is an increase of agents that target respiratory infections, such as RSV fusion inhibitors and a combination of RSV and influenza polymerase inhibitors. Those are more, I would say, classical type of targets. So we completed this analysis at the beginning of the year of 2018, and I just wanted to point out that in the first quarter of 2018, there have been a number of approvals, so 2 in the US for HIV, one that is a fairly traditional combination therapy with a new generation of integrase inhibitor. The second approval is interesting because it's the first broadly neutralizing antibody approved for HIV therapy, and that's really something that probably is going to be a precursor to more drugs of this class being approved. And in Japan, there's one molecule, baloxavir, that has been approved for influenza treatment, and that is currently in clinical trials in the US. And if I have time, I'll talk a little bit about this compound. How am I doing with time? Oh, okay, that's great. So I just wanted to conclude on the things that I just touched on. First of all, there have been 88 antiviral drugs that have been approved over the last 30 years. Most of them are small molecules that are targeting non-structural viral proteins in mainly chronic viral infections. The combination therapies have been possible because of the maturation of the clinical pipeline, both for HIV and for hepatitis C. And that actually really also translates into going from first-in-class to best-in-class treatment, so benefiting the patients. But if you look at what's really currently being tested, most of them are first-in-class monotherapies. We are observing a partial departure from the classical, I would say, antiviral drugs. So there's a number, as I mentioned, a number of oligonucleotide-based therapeutics, of cell fusion inhibitors, capsid assembly modulators for HPV, as well as host-based mechanisms and immunomodulators. So just for the last few slides, I wanted to really also address this question. And this is not going to be a comprehensive, I would say, overview of innovation in antiviral research, but I just wanted to pinpoint some stories of individual drugs that have progressed in clinical trials. Because I think behind each drug, there is actually a unique story and different circumstances on how those drugs were discovered. And I'll give two examples on influenza, because that's the type of work that my lab is also involved in. So one drug is called pimodivir. It's currently in phase 3 clinical trials. It was originally discovered by Vertex. And the way they did their screening, because it came out of a phenotypic screen, was actually very clever. So they had observed the fact that most flu drugs, things like oseltamivir, for example, they tend to be very sensitive to multiplicity of infection in vitro assays. So if you infect your cells at a high MOI, a lot of those drugs actually lose the antiviral effect. And so the Vertex scientists decided to design a screening that would purposely be run at a high MOI to actually really focus on new molecules that would still retain potency at a high MOI. And what they found were a series of kinase inhibitors, which a lot of people would just stop there and just walk away from those types of more like classical, I would say, anti-cancer drugs, because they would be considered most likely non-specific to the virus. There's a lot of kinase inhibitors that tend to come as hits in screens for antivirals, and usually they have very indirect effect that mainly involve things that can not really be developed later on. But to give credit to the people who did this study, they actually were able to identify the fact that, number one, those types of kinase inhibitors were making direct interaction with the virus. More specifically, they interact with the PB2 subunit of influenza polymerase. And number two, they were able to optimize the molecule so they could get rid of the kinase activity, and they could actually optimize the antiviral activity. And they use Vertex does a lot of structural biology, so that's one of their strengths. So they use a lot of structure-based drug design and optimization. The second example that I would like to give is going back to this drug that was approved in Japan, which is called baloxavir. So the molecule was developed by Shionogi. Also, they started from an in vitro cell-based phenotypic screen. But the strength that Shionogi had is that they had worked extensively on the HIV space. And so because of that, their library of small molecule was enriched in integrase inhibitors. And so the molecules that they found in their anti-flu assay originally were HIV integrase inhibitors. And what they were able to do is, through what we call traditional medicinal chemistry, is to re-optimize the potency to away from HIV, but more selectively towards influenza. And you can see that those types of molecules are metal chelators, so they interact with magnesium in the active site of HIV, but also in the active site of the PA subunit of influenza polymerase. So they are cap-snatching type of inhibitors, just like the previous molecule that I was telling you, even though they bind to two different subunits of the protein. Both of those molecules are first-in-class compounds. What's remarkable for especially for the PA compounds is that people in the field have tried to inhibit the cap snatching mechanism for probably about 25 years, and nobody's been able to do it until this example. And this is my last slide, so thank you very much for your attention, and I'll be happy to take questions.
Maybe I'll start with one.
Okay.
Of course, these days as a microbiologist, we hear a lot about the microbiota and how the microbiota, affects also infectious disease. My question is, has there been any study done what the effect could be of the microbiota on the outcome of a clinical trial?
Okay. So let me make sure I understand the question. I think So you're asking if the changes in the microbiome might affect the outcome of other infectious diseases. I'm not aware of any clinical trials that are looking at this specifically. I know that there's a lot right now in biotech sector looking at changes in the microbiome for other indications, all kinds of metabolic disease or even cancer. I mean, there's a lot of companies that are looking at this. But as specifically targeting infectious disease, I'm not sure about this. Maybe somebody has the answer to that.
Yeah, I'm just curious because the microbiota generates a lot of metabolites, and the metabolites could interact with the drugs and prodrugs.
Oh, I see what you're saying. Okay, from a drug-drug interaction or drug-metabolite interaction. Yeah, that's a good point. I don't have any specific answer for that though. Any other questions?
Over there.
Okay, so a couple of questions. Did you look at the source of some of these, both the approved drugs and the ones in development now? In other words, they tend to come more from academic labs originally or industry libraries. And then second question is, do compounds tend to come more from target-based screening versus phenotypic?
Okay, so the first question on the source of the, are you asking the source of the current clinical trials or the source of the approved drugs? Because those are really two different things.
Both.
Yeah, so that's a difficult thing to do because, and what we see, for example, in the pharmaceutical industry is that it's hard to trace back the story of the discovery of the original molecules. Sometimes it might start in an academic lab, then it might spin off into a small company. The small company might be bought sometimes multiple times, and then it ends up in a large pharma as a late-stage clinical trial. So in order to do sort of like all the tracing back to the original discovery, we haven't been able to do this for all drugs. I think I cited some specific examples that I know of, but we don't have this comprehensively, and it's actually even to an extent even harder to do for things that are currently on trial. And then the second question was. Oh yes. So it tends. So there's I would say different trends at different times. It's kind of cyclic. I think I would say back 10, 15 years ago, the target-based screens were very popular because they were kind of like moving away from the holistic phenotypic screen where people were frustrated with not really understanding what they were doing when they were inhibiting, in this case, viruses. But now the trend is reversing back, so people are going with better, I would say, more refined phenotypic screens. So for example, like high content imaging is very popular these days, and because there's also equal frustration in target-based screen where what you find is that oftentimes the early hits are not necessarily, don't translate in the cells. You can inhibit a protein or an enzyme, but those starting points are very difficult to translate because they have all kinds of issues with cell permeation or stability, and those things are very difficult sometimes to optimize. So it kind of changes. I think it alternates between the two.
Thank you very much.